Can you guys hear me? Hello?
Yes.
Okay, great. Thank you. I got a warning from Zoom saying that your microphone's not working, so that made me a little nervous. Okay, great. So you can hear me. It's hopefully that remains true for the recording as well. We'll see what happens. In the event that it doesn't end up recording my audio, what I'll do is I'll put together a version of this class to post as a recording, just so you know. I'll figure it out. But welcome back. Lecture 2 already, right? I'm back in Michigan, as you might see here. But yeah, we're gonna get into it. Let me pull up the slides. Thank you for a lot of you have been participating in the discussion board threads. I enjoyed reading a lot of your comments and questions. So please continue to do that for the live lectures, but also for any pre-recorded lectures so that you can get participation points. And I think pretty much everyone did the first quiz. So you kind of got a sense of what it's like. You could see they're not meant to be hard. They're not meant to be scary. They're just meant to make sure that you're attending class and looking at the lecture slides and that sort of thing. So please, please, please do them. 50% of your course grade is based on those pretty easy quizzes, right? So take advantage of that. Don't drop the ball on those. Today's lecture is obviously live on Wednesday. That lecture will also be live. Next Monday and next Wednesday, those lectures will be pre-recorded. I'll put reminders in Canvas so that you know that. Again, I'm saying it now, but I'll remind you of that again before those classes. And obviously, if it's a pre-recorded lecture, we won't be meeting in a Zoom, right? There will be a link for you to click on and you'll be able to watch the recording. If there's a question, if we attend the live lecture, do we have to, oh, that went away. Do we have to post in the discussion thread as well, or is that only for pre-recorded sessions if we do not attend the live one? I would, if you attend, so there's attendance and then there's participation. For the live lectures, Zoom generates attendance for me. So I can see who attended and who did not attend the live lecture. For the pre-recorded lectures, obviously, any attendance for that will be strictly based on the discussion board threads. If you participate in the live lecture, like via the chat box or something like that, I can give you participation points there too. It can get a little tricky though sometimes because so I would recommend, just so we're like even if, how about this? If you attend the live lecture and you say something in the chat box, then go to the discussion board thread and be like, oh, hi, like, yeah, remember I asked that question about whatever during the live lecture, just so I get reminded. Or I could also go into Zoom settings to save the chat box, but I don't have that set up as of right now. Okay, I'm gonna share my screen because I always say the sooner that we get started, the sooner we finish. The sooner that we finish. So let me pull this here, come on. We have a decent amount to cover today. I'm going to try to get through it as quickly and painlessly as possible. Obviously, this will be recorded. So if there's a part that goes a little quickly, you can always rewatch it later. So hopefully everybody right now, can see the slide, right? Some of you have already been emailing me, so that's great. If you have any other questions that you don't feel comfortable asking them in the chat box, or yeah, you could always feel free to follow up in an email with me. I'm pretty good about turnaround time. Okay, lecture 2. All right, last time we reviewed this syllabus and a little bit of an introduction to the course, including how plants are important to us, how we classify plants, and life on Earth. Today, we're going to get into a little bit more biology. It's going to be a little bit more unfamiliar terms, new words, perhaps new concepts. So I'm saying that to be at the beginning because I don't want you to feel stressed out. It is a biology class, so there are going to be words that we encounter that are brand new to us, most likely. To make you feel less stressed out, though, I will say that there's a lot of repetition in this class. So you might today see a big scary word that you have never seen before, but then you're going to see that big scary word a bunch more times. So if today you're like, ooh, this is a lot of new words, don't worry. So we're going to go through tissues and stems. We're going to talk about a little bit how plants grow, and the tissues that are responsible for that, which are called meristems. And the meristems will make tissues, which we will see in today. And then we'll talk about stems, in particular dicot stem versus monocot stems, and then some examples of plants that have variation in stems, which allows those plants to be adapted to do different functions. All right, before we can kind of talk about the stem, we kind of have to talk about the plant body in general. Like, oh, you have a question? Here, let me make sure didn't miss anything in the chat box. No, we're good. Okay. Yeah, so like plants have bodies. You may not think about that, but plants have bodies. You're an animal, you're a human, so you have body parts. You have hands, you have arms, you have a head, right? But plants also have body parts. And obviously they're quite different than the body parts that we have, right? Because they're plants. So this is a handy slide that kind of just is a little roadmap of some of the general parts of a plant. In particular, what I want to draw attention to on this slide, and what's a good color for this? Let's do dark blue. Most plants grow in the ground, right? We will learn going forward, there's exceptions, right? Some plants grow in water and float, some plants grow on other plants, but most plants grow in the ground. And for those plants, it's easy to see these two systems. Well, maybe it's easy to see one of them. But all plants have a root system and all plants have a shoot system, right? For plants that grow in the ground, the root system is usually below the surface of the soil and the shoot system is usually above the soil, right? Some familiar things that maybe you already knew a plant have, right? Like a leaf. Leaf is part of the shoot system, whereas different roots, right? So here you can see a tap root and a lateral root. We'll learn more about roots in the root lecture, are part of the root system. This plant also happens to be part of the flowering plant. So obviously here there's a nice little flower. But today we're going to talk about some things that show up on this slide. For example, apical bud, because we're talking about stems today, and we'll go into it in more detail. But basically this apical bud, here's one. These are buds that are at the tips of stems. And so as the stem grows, it's that bud that's going to be making the new tissues. Okay, so we'll talk about that a little bit more in detail here. Okay, so what does the root system do? The root system anchors the plant. Now, obviously, if you're a plant that floats around in the water, the root system isn't doing that. But for most plants, the root system is anchoring the plant, whether it's anchoring it into the soil or anchoring it onto a rock or anchoring it onto another plant. In addition to anchoring plants to a surface, roots also absorb water, and that's probably the function you're most familiar with. If you have houseplants, you water the soil because the roots are down there, right? And that's where they absorb water. They absorb water and minerals down there. Minerals, for example, calcium, right? Okay, because that's in the soil. Then you have the shoot system. The shoot system is composed of the stems and the leaves. The stems serve to raise the leaves, which are the photosynthetic organs. So the leaves are what makes, the leaves are where photosynthesis occurs. And you need the sun to have photosynthesis occur. And so the stem's function is to hold the leaves up closer to the sun. And then on stems, you also have reproductive structures attached. And if you're, for example, a flowering plant, you would have flowers, those reproductive structures attached to the stems. But not all plants are flowering plants, so there will be other reproductive structures we come across as we go forward in the course. All right. And then lastly is this thing called a vascular system. So you and I have a vascular system. We have veins and capillaries and arteries, right, that move blood throughout our body. That's our vascular system. Plants don't have that. They have two other types of tissues, one of which is called xylem tissue, which is right here. You say xylem like xylophone, right? Xylem tissue and phloem tissue. That pH is a like phone. So xylem tissue and phloem tissue. Xylem tissue is going to move water and minerals from the roots up to the leaves. Phloem tissue is going to move the products of photosynthesis, which is a fancy way of saying sugars. Phloem tissue is going to move sugars from glucose from the leaves down to the roots and other parts of the plant, right? Okay. We'll talk about these in more detail as we go ahead. But these are the basic body parts of a plant: a root system, a shoot system, and a vascular system. Okay. Now that we know the basic parts of a plant, we're going to talk a little bit about how those parts grow, because just like you and me, plants have to grow. First thing I want to point out is that in plants, and probably in us too, but I don't really know as much about it in humans, but in plants, there's something called there's two types of growth in plants. There's primary growth, which is to lengthen, to get longer. And then there's secondary growth, which is to get wider. So what you could think about, and I have it in this like drawing here, or I guess pictures. I'll change colors to, I guess, red for this side. So you see here on the left, there's this little tiny tree, right? It's super tiny, and the roots need to get longer, the stems need to get longer for it to grow in length. And so, for example, maybe here there's a stem, and the stem wants to get longer. Oops, told you I'm bad at annotating. But as that occurs, that's an example of primary growth. Whereas on the side here on the right, you see this big tree. Look how thick the stem of that tree is compared to the stem of this tree. So the way that parts of plants get thicker is via secondary growth, which serves to increase the width of the plant body. And I have a question. Is there a particular time span for each growth? That's a great question. So it will depend on the plant, but for what it's worth, all plants have primary growth occurring all the time on some part of their plant body. So primary growth, that increasing in length of a root or increasing in length of a shoot, that's happening all the time in all plants. Secondary growth, or increasing the width of a plant body, only occurs in some plants. For example, this woody tree. But for example, a grass, like a blade of grass is not going to ever really increase in width. It's only going to increase in length. So grass doesn't have a lot of secondary growth. In terms of the timing, it really depends on the lifespan of the species. So for example, in this woody tree example, you know, this tree over here is probably, I don't know, 10 years old, give or take. Whereas this tree on the right here is like probably 100+ years old. So yeah, good question though. Okay, that's what I was just saying. All plants have primary growth. All plants have primary growth. No exceptions. Only some plants though have secondary growth. All right, Now that we know a little bit, just a little bit about the types of growth, right? We know that there's primary growth, which is for lengthening, and we know there's secondary growth, which is for widening. We'll cover just a few different types of plants. I got a question, what can prevent a plant to fully completing primary and secondary growth? So some plants, that's actually what we're going to cover. So with this question, what can prevent a plant to fully, that's kind of what we're going to cover now with the different types of plants, Baba. What hinders growth? No matter what I do, all my plants die. There's a lot that could be going on, Sharonda. And if you want, send me photos in an email and I could help you troubleshoot. Do plants grow in the reverse order? Like, do some plants get wider before they get longer? So on certain plants, certain parts of a plant may be getting wider. Like, for example, the trunk of a tree might be getting wider while the tips of a stem are getting longer. So if you're a plant that has both primary and secondary growth, you could be having both happening at the same times. Okay, so our herbs, so let me tell you, before we go into all these questions, let me tell you quickly what the types of plants are, because I have a feeling that some of your questions might be answered in these next slides. For example, are herbs different than aromatics? So herbs are a type of plant. And you've probably heard of herbs as something you cook with, right? Cilantro, parsley, basil, dill, right? Those are things that you think of as herbs. And they are herbs. But in botanical sense, or in plant world, right? Herb just means that you're a type of plant that never gets woody. You never get woody. So herbaceous means non-woody. So the grass that I mentioned a couple seconds ago, that's an example of a plant that never gets woody. You don't have woody grass, right? You have soft grass, soft herbaceous grass. So, yeah, so all those aromatic herbs that you use for cooking are herbs, but that's not all herbs. Herbs are also anything that's non-woody. So you could see here, there's a lot of flowering plants, for example, tulips, daffodils, those kind of spring blooming plants are all examples of herbs as well. Okay, then you have shrubs. Shrubs are capable, oh, and importantly, herbs only have primary growth, right? Herbs don't ever get wood, so no secondary growth for the herbs. Shrubs, on the other hand, are things that are kind of shorter than trees, right? But can make wood. So for example, this rose bush that I have shown here, right? Rose bush gets a little bit woody. And so that's an example of a shrub. And shrubs have both primary growth and secondary growth. So shrubs can produce wood. And lastly, trees, right? So trees have both primary and secondary growth. We should probably already know, without even taking this class, that trees can make wood. So those are the different types of plants. Okay, now that we know herbs only have primary growth, shrubs have primary and secondary growth, trees have primary and secondary growth, we have to talk a little bit about what primary and secondary growth even means. So remember I circled that apical meristem, or that apical bud, back a few slides ago? So the apical buds, or apical meristem, on this slide it says terminal, but don't worry about that. It's still apical. It just means it's apical, terminal, however you want to call it. It means it's on the tip, the apex, right? It's on the apex of the stem or the root. And so that's where you're having lengthening occur, right? If you're a bud right here, you want to get the branch, sorry, the stem longer, right? So you're going to have primary growth so that the plant can grow, so the stem can grow longer. So apical meristems, which are at the tips, are responsible for primary growth. Oh yeah, here, I will show you. Okay, so let me stop share for a sec. And let me pull this up real quick, just to show you in real time. All right, so I'm gonna go here and put it here. I know you probably can't see it yet. 1 second, I'm gonna wait to see if there's an ad, which of course there usually is. Hold on, letting this ad go by. Okay, good for you, ad. All right, now I'm gonna share. Screen. All right, so share screen. Sunflower. Here we go, share. So you should be able to see the screen. I'm gonna mute it because you don't need to hear anything. Let me know if you can't see the video. So here is, we're gonna watch a sunflower, which is growing from a seed all the way through making a flower. But what I'm trying to show you here is, so look, what's happening right now. The stem of this sunflower is lengthening, right? Sunflowers, so look, I'll pause it right there, pause. Where's my doodad here? I'll stick with red. So right here is where that apical bud or apical meristem is going to be. And it's going to, there's going to be cell division and things, processes that are happening at that bud that are going to allow new tissues to be made during primary growth of lengthening. So let's see it go. Oh, I got to get out of this. Here we go. All right, so look. So at that apical bud, you have lengthening of the stem. And as the lengthening of the stem is happening, other things are happening, like new leaves are being made. But notice the stem getting longer and longer and longer. You can't see it, but under the soil, the roots are getting longer and longer and longer, right? And that's your primary growth. I could literally watch time elapses of plants growing all day. If anyone ever tells me plants don't move, I just walk away. Plants are moving all the time. They just move a little slower than us. So look, so now this sunflower's already gotten to the point now where it's starting, the stem is starting to make some of those reproductive structures. So that thing at the tip now, this is the new flower, this here, this, I'll use a different color, right? This is a flower that's going to be emerging. Because remember, the stem makes both leaves and the vegetative structures, right? So there you go. You see that little movement there, that kind of like little head banging movement? We'll learn more about that in the physiology lecture. Yeah, there's actually a name for that. And there you go. Look at that. Wow. Sunflower, right? Sunflower. Okay. If you want to watch the whole thing, you can. You'll have access to the link in the lecture slides. All right. Let's share this back. And here, give me 1 second. Close this for a sec, make this big. Quick question in the chat box of why are the leaves dying? Right, so some of those lower leaves die because they're no longer needed. As new leaves are being made at the top of the shoot, the lower leaves are no longer to really be able to grab sunlight because they're being shaded out. And so the new leaves, which are much bigger and newer, I think it's in the details of that link. I don't remember what the actual time is of that specific time lapse. I include a lot of time lapses in this class. But yeah, so that's why the leaves are dying, because those leaves are no longer needed. Okay, so primary growth is at those apical meristems, right? And so what you should know is that the primary growth that's happening at the apical meristems, and I'll stick with red for that, right? Primary growth at the apical meristems. But the apical meristems, or apical bud, are the same, whatever you want to call it, are going to give rise to the primary meristems. Primary meristems. What does that mean? Let's look. Okay, so here, but so you're oriented. Just so you're oriented, right? So primary growth is where? The apical meristems. Where's the apical meristems located? At the tips. So right here is one. Here's another one at the tip of that stem, right? So we're going to look under the microscope at that tip, right? That's what this is showing you. So we're looking at a cross-section of, well, it's actually a longitudinal section. Don't worry. We're looking at this stem tip under the microscope. And what you can see, look, apical meristem. So apical meristem is right here. And the apical meristem is going to be is the apical meristem is made out of cells. And maybe you've heard the term stem cell before, right? So like a stem cell is a cell that can grow and divide into different tissues, right? So like a stem cell could grow into like skin tissue, or it could grow into lung tissue, or it could grow into bone tissue. So maybe you've heard the term stem cell before. Well, that's why we call these meristems, because the cells that are in that apical meristem are basically stem cells. And so they're going to grow and divide. And because we're talking about a plant and not a human, they're going to grow and divide into primary meristems that lead to the parts of the plant body that I've already talked about, right? Like a leaf. So you just stick with me here, right? So primary growth is occurring at the apical meristem, and the apical meristem is dividing and giving rise to primary meristems. The names of those primary meristems, which again, we will see many, many, many times, are protoderm, procambium, and ground meristem. Those are the three primary meristems that the apical meristem makes or gives rise to. Make and give rise to are the same thing. Okay. See, when you're looking at the slides later, it's color-coded for you, right? Okay. It is the same thing. This is an actual microscope slide. This is a drawing. This is a drawing. And what do I, yeah, so just a different view of it. I will say though, I just noticed that, so here, like the procambium, right, is purple. And sorry, the procambium, right, procambium and ground meristem are kind of more toward the middle of the stem as the apical meristem makes it. Protoderm is going to be like kind of like up here. That doesn't really matter so much. I'm just trying to show you different ways of looking at the same thing, right? This is the same as this. This is a drawing, right? This is also a different drawing showing the same thing. So, you know, here I'll do a little, right? So this apical meristem is growing and dividing. And as it grows and divides, it's making, oh man, sorry. I'm gonna try really hard here. It's making procambium. Sorry, procambium and protoderm, which are kind of located in the same spot. And then it's making ground meristem behind it. Yeah. Don't worry too much about that. What I want you to put in your head is that you have apical meristems that make three primary meristems, and the primary meristems are procambium, protoderm, and ground meristem. Okay. Because those primary meristems are important. They're going to create the parts of the plant body. Do succulents also grow like this? Yes. Yes, they do. So those primary meristems are going to grow the parts of the plant body, right? They're going to grow the stem, they're going to grow leaves, they're going to grow roots. And in order to do that, those primary meristems are going to make what are called primary tissues. And don't worry about these too much right now, because we will come across them a bunch of times. And if you happen to be in lab, you'll also come across the terms in lab. If you're not in lab, don't worry, we'll come across it plenty of times here. But primary meristems make primary tissues. There are some simple tissues that are made up of one cell type. There are some complex tissues that are made up of many different cell types. Yes, okay, good. Right, so we're going to go through these so you are familiar with them. So, and I have a nice schematic at the end that kind of sums this all up too. So you got those primary meristems, right, which were called the procambium, protoderm, and ground meristem. Those primary meristems are going to make primary tissues. Simple tissues are parenchyma, cholenchyma, and sclerenchyma. Yes, those are real words. We'll come across them a bunch of times, don't worry. And then there's complex tissues that are made up of a lot of different cell types, And some examples of complex tissues are the pith, the cortex, the epidermis, the xylem, and the phloem. Okay, so this is some of the simple tissues I just mentioned, simple primary tissues. This is parenchyma under the microscope slide. So each of these cells, I'll use black. Each of these round things is a parenchyma cell. And this whole thing here is parenchyma tissue. Parenchyma tissue is only made up of parenchyma cells, so we say that it's a simple tissue, simple primary tissue. And as you can see on the bottom, parenchyma tissue is the most abundant type of cell found in plants. So if you're looking at a plant under the microscope slide, there's a really good chance that you're seeing some parenchyma cells because it's the most common type of cell in plants. Here is that sclerenchyma. So likewise, since it's a simple tissue, so this is one sclerenchyma cell, this is one sclerenchyma cell, this whole thing is sclerenchyma tissue, but sclerenchyma tissue is only made up of sclerenchyma cells, so it's a simple primary tissue. These cells are kind of rubbery, so it says pliable and strong, and they basically provide flexible support for growing the mature organs. So for example, if you've ever eaten celery and there's those weird strings that get caught in your throat, right? Those weird strings are sclerenchyma tissue. That's probably the most familiar example that you've ever encountered. Okay. And then lastly, there is this other simple tissue called sclerenchyma. Sclerenchyma is made up of one cell type. Sorry. So again, just to hit this home, right? I think this is like, actually, I think this is a close up. Oh, right. Yes. Sclerenchyma tissue is simple because it's made up of only sclerenchyma cells. But there are two types of sclerenchyma cells. There are ones that are called sclerids, which sounds like a Harry Potter thing, and fibers. And so if you're a sclerenchyma tissue made out of sclerids are these weird star-shaped kind of tentacle-looking things. So this is an example of sclerenchyma tissue made up of sclerids. And then this up here and this here, etc., is example of sclerenchyma tissue that's made up of fiber cells. Okay. Any relation to the strings on a banana or the fleshy parts of a mango? Both of those are parts of fruits. I'd have to look into that. But probably not so much the banana. They may have been, well, yeah, I don't know. Okay, yep, so next, I think, oh, right. Oh, yeah, so just to give you an example, so like the sclerid type, the sclerid cells of sclerenchyma tissue are kind of like gritty. So if you've ever eaten a pear and it kind of feels like you're kind of like chewing grit, right? Or like sand when you're eating the pear. That's because of those sclerids, which have that kind of a crunchy vibe to them if you eat them. Fiber cells, on the other hand, are things that you could use to make certain textiles. So for example, banana, bamboo, jute, other examples, sisal, hemp, those plants have a lot of fiber cells in them, which is why we can use them to make like textiles and other material products. Okay. Oh boy. All right. So that was the simple tissues, right? There's simple tissues, parenchyma, parenchyma, and sclerenchyma. And then there's complex tissues, right? Pith, cortex, epidermis, xylem, and phloem. So we went through the primary tissues that are simple. Now we're going to go through the complex ones. So if you're a complex primary tissue, it means that your tissue is composed of many cell types. So for example, xylem. Xylem is a tissue And xylem is composed of multiple cell types, tracheid cells, and vessel element cells. We'll learn more about that in detail later, but like, for example, these are tracheid cells, and these are vessel element cells. Just trying to get you familiar with the idea of like complex. Similarly, similarly, phloem. Phloem is xylem moves water. Remember I said at the top of the class that xylem moves water from the roots to the rest of the plant, including the leaves. So you can see like the xylem cells kind of look like straws a little bit. Phloem cells, or phloem tissue rather, is composed of a couple different cells. We'll talk more about them in detail, I think, during the plant physiology one, but you have sieve tube, companion cell, sieve plate. Don't worry about that too much right now. I'm just trying to show you that xylem is a complex primary tissue, right? Phloem is a complex primary tissue made up of a lot of different cell types. Epidermis is a complex primary tissue. The epidermis is the outside of any part of the plant. So if you're looking at a stem, which we'll go into this in detail more later, but the outermost tissue, we have epidermis too. Our skin, the top of our skin, this is called epidermis as well. And our skin has different cell types on it too. Some of our epidermis makes like hairs, right? Some of our epidermis doesn't make hairs, right? Some of our epidermis makes like, has glands, all different cell types. Yeah, so epidermis is another example of a complex primary tissue. Because, so look, so this here, I should have maybe been more clear. This is a cross-section of a leaf through a microscope or under the microscope. And I'll use black. So a leaf, imagine my hand as a leaf. A leaf has a lower surface and a leaf has an upper surface. And so there'll be epidermis on both. So this here is the epidermis. This here is the epidermis on the bottom of the leaf. Just to show you what this slide is showing you. And so you might, when you look at that, you might say to me, well, Rebecca, this looks pretty uniform. Like it does not look like multiple cell types. That looks like a single cell type. Yeah, it kind of might look like that, but we know it's not the case because there's a lot of different types of epidermal cells that make different things. So some of them make hair, just like our epidermis. Some epidermis cells make hairs. For example, this on the left, these weird, like kind of scary looking green structures are hairs on the leaf, known as a trichome. Other cells of the epidermis might make oil glands, which are these big, like mushroom looking things on the right-hand side. But the important thing for you to understand is just that there are simple primary tissues and complex primary tissues. Xylem, phloem, epidermis are examples of complex primary tissues. The purple on the left side, there's a question in the chat box, is also oil being secreted from other epidermal cells. Okay. All right. This part's going to be a little, this next few slides are probably going to be a little confusing. So I will just say that from the get-go. And so for this, when you have the lecture slides later, you might want to kind of really take your time on these next few slides. But basically, I'm going to try to show you when I said you have those apical meristems, right, that are at tips. And those apical meristems make the primary meristems, which are called, ground meristem, procambium, and protoderm. And that those primary meristems are going to produce primary growth tissues. Some of those tissues will be simple. Some of those tissues will be complex, right? But they're still primary growth tissues. Okay, so for example, and notice the color coding here. right? Notice the color coding here. So look, here, I'll do this. Okay. We didn't talk about it in detail yet, but the ground meristem, which is a primary meristem, makes the tissues that are inside of the stem or inside of the root or inside of the leaf. So in this example, we're talking about a stem, and inside of a stem, the ground meristem is going to make these tissues, the pith and the cortex. So that's what the ground meristem is going to make. What's next here? Yep. We didn't talk about it in detail, but the procambium, which is a primary meristem, right, is going to make primary tissues that are called primary phloem and primary xylem. And then lastly, you have, and notice the location. Look. Where's the procambium? It's in this little thing, right? Each of these little things has procambium in them. And where's xylem and phloem when it gets made? Well, what do you know? Same location, right? Same location, primary xylem, primary phloem, because it was made by that procambium. And then lastly, we got the protoderm. The protoderm, it's very useful that protoderm has the word derm in it, because the protoderm makes epidermis, right? Epidermis. So that's helpful. Okay. Oh, good. This is good. Okay. So, you know, it's one thing to look at a confusing thing like this. Let's think about it in terms of the body parts of a plant, right? Body parts of a plant. So, remember, you have protoderm, right? Which is the primary meristem, and it makes the epidermis. And where does the protoderm make epidermis? It makes epidermis on the leaf, it makes epidermis on the stem, and it makes epidermis on the root. So, just for simplicity, top here is a cross section through a leaf. Here is the upper epidermis. Here's the lower epidermis. Middle picture here is a cross section through a stem like that. And you have on the outside, that stem, the epidermis covering the outside. Lastly, picture here on the bottom is a root. If we did a cross section through a root, the outermost layer would be that epidermis. And so in all cases, that's made by the protoderm. Next, we have ground tissue system. Remember, that's made by your ground meristem, right? So as we learned on this slide, ground meristem is going to make pith and cortex. So pith and cortex are some examples of this ground meristem, or some example of this ground tissue. Let's start from the root. If we do a cross section through a root, the ground meristem is the cortex and the pith. So pith is not labeled on this slide here, but in this root cross section, all of this part is cortex. If we did a cross section through the stem, likewise, all this inside section is cortex, ground meristem. However, on a leaf, the ground meristem produces a tissue called mesophyll. So if it's in a leaf, all this stuff in the middle we call mesophyll. And interestingly, mesophyll means middle of leaf. Meso means middle, phyll means leaf. So mesophyll is the tissue that's located in the middle of a leaf. And then lastly is your vascular tissue system. So this is stuff made by who? Made by who? Made by the procambium. Remember, procambium makes the primary phloem and primary xylem. And no matter where you are in the plant, whether you're in the leaf or the stem or the root, the primary xylem it's always called primary xylem and primary phloem for primary growth. So in the leaf, primary xylem and primary phloem are going to be located in this area, which is the vein of a leaf. In a stem, in the center of the stem, you'll have primary xylem and primary phloem. And in a root, you'll have primary xylem, primary phloem. And all of those were made by the procambium, which is a primary meristem. Again, I know this is a lot of new information, so just try to be in it today and learn it, hearing all these new words for the first time. You'll have the lecture slides and you'll have the recording to go back and kind of listen again where you got a little lost, right? Okay, this is a summary, kind of. So you have that primary growth at the apical meristem, right? What's primary growth for? Lengthening, right? At that apical meristem, you're going to have, the apical meristem is going to make primary meristems, right? What are the primary meristems? The primary meristems are protoderm, ground meristem, and procambium. And each of those primary meristems are going to give rise to primary tissues. Protoderm will make epidermis. On the last few slides, we saw where that occurs in the plant. Ground meristem is going to make the ground tissues, which is parenchyma, parenchyma, sclerenchyma. Those make up the pith and the cortex. And lastly, procambium is going to make your primary xylem and primary phloem, right? So this is the schematic I was telling you about, like apical meristem makes, apical meristem makes all of those ground meristem, right, primary meristem, and each of those makes their own primary tissue. Clear drawings. Okay. Okay, that's primary growth. There's also secondary growth, which I mentioned at the top of the class. Not all plants do, right? So like herbs don't do secondary growth. Shrubs and trees do secondary growth. Secondary growth is for widening. And because secondary growth is for making things wide as versus long, they are not in apical meristems. They are in what are called lateral meristems. That's where secondary growth occurs. So just to compare, right? Primary growth is occurring at the tips. Where? At the apical meristem. Secondary growth is occurring not at the tips, in other parts of the plant that are called lateral meristems. And I'm doing this because in both cases, there are two lateral meristems, the vascular cambium and the cork cambium. And in both cases, they are shaped like a ring, which is why I was doing that. Because when I think of lateral meristem, I immediately think of a ring shape. The vascular cambium is going to give rise or make the secondary xylem and secondary phloem. And the cork cambium is going to give rise to what's called cork and phelloderm. Again, I know it's in a lot of new words, but we're going to have a lot of repetition. And so this is just a reminder there with that arrow, right? Secondary growth, which I color code in blue, is for widening. So this is a helpful slide that you might want to just like print out or have handy, like take a screen when you get to pull up the lecture slides, maybe take a screenshot of this slide because it is a nice overview. Just to recall, just to give you here, so primary meristem, apical meristem makes what? Makes these primary meristems. What do those primary meristems make? Different primary tissues that we just covered, right? But eventually, so primary growths are happening at the tip. But as the tip gets longer and longer, it's kind of like leaving stuff behind it, right? Like as it's growing longer and longer and longer, like this part isn't at the tip anymore. Now the tip's up here, but it did have the tip there. So there will be a shift. For plants that are able to do secondary growth, there will be a shift from, and I think there was a question in the chat box that was kind of asking about the timing of this. As something gets older, it'll start off by getting longer, but then also needs to get thicker. You can think of a human, right? I know for me, when I was growing as a kid, right, most of my growth was to get longer, right? But now that I'm a grown up, it seems like most of my growth is to get wider, right? So same thing happens with plants, right? Like same thing happens with plants. All right. The reason why I'm showing you this is that, so eventually, and blue is not going to be great, but like eventually you're going to end up with these two secondary meristems, right? These lateral meristems, cork cambium and vascular cambium. Actually, I think I used yellow on that, but here I have it highlighted here. Yeah. So the color, so apical meristem, apical meristem leads to lateral meristems because you have primary and secondary growth. Here, color-coded, you have those primary meristems in purple, protoderm, ground meristem, procambium. And you have those secondary meristems in yellow, aka lateral meristems, cork cambium, vascular cambium. And cork cambium and vascular cambium are each going to be making, as I said on the previous slide, secondary tissues, right? Secondary tissues. Cork cambium will make cork and phelloderm. Vascular cambium will make secondary xylem, secondary phloem. Yeah, let's go back. Do plants ever grow at an accelerated rate and develop any sort of stretch marks? Yes. The simple answer is yes. Yeah. Okay, so I know this is kind of like, why does she keep showing us these things? Because it's going to be useful later, like with the color coding. I think it'll help you. So you might be looking at this, and I told you before, right, that the apical meristem is making these primary meristems, right? That's where we get the primary meristems. The apical meristem makes them. But how do we get these secondary meristems, right? Where do these lateral meristems even come from? How did we get those? And so the way we get those is that those lateral meristems are actually going to be made by some of these primary tissues. You could see the arrows here. I'll use pink, right? So the cortex will grow and grow, and then eventually the cortex will create the cork cambium. And likewise, don't worry about some of these other terms that I'm not mentioning here. But the vascular cambium is made, don't worry about these scary words. But the vascular cambium is going to be made from parts of the ground tissues and parts of the vascular tissues. In this class, you do not need to know the details of that. Just know to simplify. What makes primary meristems? Apical meristem. What makes secondary meristems or lateral meristems? The primary tissues. Okay. And then, as I already said a couple times now, the lateral meristems make these secondary tissues: cork, phelloderm, secondary xylem, secondary phloem. Okay. And this is a term in particular, if you're in the lab, you might see this term, periderm. Periderm is just a collective term that refers to the cork, cambium, cork, and phelloderm collectively. So if you see the term periderm, that's what that refers to. You see the little asterisk like next to cork, cambium, cork, and phelloderm, because you can collectively say the periderm, and it would mean all of those things. All right, so now we'll just do a little bit of a visual overview of primary and secondary growth. So first we'll start at the tip, right? And what's happening there? You have the apical meristem that gives rise to the to the primary meristems, right? Procambium, protoderm, and ground meristem. And those will give rise to those primary tissues, right? Epidermis, cortex, primary phloem, primary xylem, and pith. Eventually, though, parts of those primary tissues, right, will make these lateral meristems, and then those lateral meristems will go on to make the secondary tissues. So just do I color code it? I don't. Just to give you a little thing here, right? Remember, primary growth is for lengthening, so that's where that red arrow is showing you going up and down. You might be wondering though, where are those, and I'll use black because it'll show up better, but for those secondary or lateral meristems, right? Cork cambium and vascular cambium. Remember I told you that they're in a ring? So look, the blue line, this is going to be hard for me to do, but the blue line here, it goes all the way around, is the cork cambium, and the red line in this drawing, oh, you see, they're in rings. They're in rings. Okay. Primary growth, secondary growth. Okay. This is kind of showing the same visualization of a transition from a primary growth to secondary growth. So here you go. This is what we saw earlier, right? You have these three primary meristems, right? Ground meristem, procambium, and protoderm. And those are going to give rise to primary tissues. What are the primary tissues? Epidermis, pith, cortex, primary phloem, primary xylem. And then eventually some of those primary tissues are going to give rise to the secondary tissues via those lateral meristems. And do I color code it here? Let's see. No. So let's use yellow. So vascular cambium. Actually, well, hold on, I gotta move this. Cork cambium isn't here. Cork cambium isn't labeled here, but cork cambium would be like around here once it's there. And like I said, vascular cambium is the one toward the middle. Rings, right? Rings. And then eventually, for example, in a tree, if you were to look at a tree trunk, like if you were to cut a tree down and look at the stump, when you get to that point, that stump only has secondary tissues, So if you were to look at the tip of that tree, you might see some primary tissues like epidermis, primary xylem, primary phloem, cortex, and pith. But if you were to look at the bottom of that tree and that, in the big trunk, at that point, you're only seeing these secondary tissues, right? See, eventually there will be no primary tissues remaining in the stem, only secondary tissue. And what are the secondary tissues? Right? Cork and phloderm and secondary xylem and secondary phloem, right? Secondary, no, I didn't do it. Sorry, I'm not trying to make you dizzy. I'm making myself dizzy. Okay. This will be just helpful for you later, but like, remember I said that go back here, sorry. Right. Remember I said like the vascular cambium, right? So there's two lateral meristems, right? There's the cork cambium and the vascular cambium. The vascular cambium is going to make secondary xylem down here and secondary phloem. Because the vascular cambium is in a ring, there's an outside part of that ring, like out, and then there's an inside part of that ring. All right, close your eyes. I'm not trying to make you dizzy. Okay. So when the vascular cambium is making the secondary xylem and secondary phloem, the secondary xylem is always going to grow toward the inside of the stem. So let's see here. Right. So picture like right here. Whoops. Here. Picture right here. Like where I'm putting this yellow line to be that vascular cambium. And the secondary xylem is always going to grow toward the center of the stem or toward the inside of that ring. And the secondary phloem is always going to grow toward the outside of the ring of the vascular cambium. And so it says there. Yep. So this is it in a real microscope slide. So if we were, so if you were to look here, this is a cross section through a stem. You could see some of the primary tissues we already talked about, right? So for example, On the outside of the stem is the epidermis. On the inside of the stem is all the cortex and pith. And then you notice these little areas that look kind of different. These little areas. So for example, let's take this one and look at it up close, which is what B is showing you here. Let's use the same color. So like here, look, vascular cambium grows in a ring. Can't see it all. I can't draw the whole ring there. I'll draw it here. Vascular cambium. That's like in a ring. And secondary xylem is going to grow toward the inside of the stem. Secondary phloem is going to grow toward the outside of the stem. Like that. All right. Once the stem is mature, these are what you're kind of more familiar with, right? So like a woody twig or a woody branch. Notice though, there's still this terminal area here, right? So primary growth is still going to occur there as the branch gets longer, right? But notice already, like down here, how thick the branch has gotten because secondary growth has already occurred. The reason why I'm showing you this is because depending on where you cut through this branch or twig, you might see only primary tissues, or you might see only secondary tissues, depending on where you cut it. So if you were to cut right through here and look at it under the microscope, you would probably only see primary tissues. Whereas if you cut down here and looked at it under the microscope, you'd probably see mostly, if not all, only secondary tissues. So I'm trying to get you to think about this like transition period. Yeah, okay. I know this is hard. We're almost there. We're getting there. We're almost there. Okay. When you see a tree trunk, right, you should just be thinking to yourself now, because you're an expert now, that that's secondary growth, because you're seeing something on a plant that's super wide, something on a plant that's woody, right? So tree trunks are an example of secondary growth. Check out this tree. Whoa. Whoa, right? Here, I'll use blue. Look how wide that trunk is. So this tree has abundant secondary growth, right? Especially down there. But even, look, even on these branches, right? Look how wide this branch is, right? This branch is not so wide, but it's still pretty wide. It's way wider than this up here. And at the very tippy tips of the branches, you'll probably still have primary growth occurring as they get longer, right? Okay. And there's a question, how about the side? Not really sure what you mean by that. So if you could clarify.
Professor, you know the branch is trying to hug the tree. So is that the first or the secondary? The branch is just coming out and branching out. That's the one. Yes, you're just clicking.
Yeah. So like all of these, anytime you see wood with your eye, like secondary growth has already occurred. Because the epidermis, for example, is like, think of like a green stem or like a green leaf, right? Epidermis is usually green. So by the time you see like a woody structure, you're talking secondary growth, but also look at how thick, right? Like this is very thick. So we already know that secondary growth has occurred and it's gotten wider. All right, oops.
Thank you.
Yeah, no problem. Okay, so to kind of sum it all up again, secondary growth is via these lateral meristems. What are their names? Vascular cambium and cork cambium. What does the vascular cambium make? Secondary xylem, secondary phloem. What does the cork cambium make? Cork, aka phellum, and phelloderm. And just so you know, what we call wood, right? Wood. If you have a wood floor, if you have a wooden door, if you have a wooden bench, right? If you have a wooden ruler, a wooden table, all of those things are made of only secondary xylem. So what you and I call wood, like right now my computer is on a wooden table, right? Wood, if you ever go to like a store and buy 2x4s, right? Or a sheet of plywood, right? Well, I don't know so much about what's in plywood. Maybe there's other things there, but when you think wood, like a 2x4 of wood, that's all only secondary xylem, just so you know. Okay. Look at this. All right, so yeah. So this is an example of only secondary tissues. This is kind of what I was saying before here. Like if you were to do like, if you were to cut through here and look at that, you wouldn't see any primary tissues anymore. No, you'd see something looks like this. And so what are we seeing on something like this? I'm gonna have to use some different colors here. You would have here, I'll use yellow. Yeah, I'll use, it's gonna be hard for me to draw. Can I do a shape? Yeah, ooh, I could do a shape. Okay, let's see if this works. All right, all right. Can I make it bigger now that I drew it? No, I gotta get better at my shapes. All right, nope. Here, I'll try one more time. Let's try one more time. Oh, it's so not good. One more time. Nope. Okay, forget the shape. My point is that I was trying to show you that, like, I remember I was saying wood before. Like when you get to this stage and you're looking at a slice through a tree like this, most of what you're looking at is secondary xylem, aka wood, right? So I'll use white, but like everything from all of this, oh boy, all of this stuff here in the middle, that's all secondary xylem only, right? The secondary phloem and the two lateral meristems are all going to be located basically like right up here. So everything else inside of this old trunk is secondary xylem, aka wood, Okay. This is the same thing, but like a microscope slide. So here's what it looks like in real. Here's a labeled version. Just to use the same colors. I know you're like, not again with these colors. Yeah, so look, vascular cambium. Where's our cork cambium? Cork cambium, remember they are in rings. So like vascular cambium is going all the way around. Can't draw the whole ring here, but yeah, all the way around. Cork cambium is also in a ring. So cork cambium is going all the way around too, right? All the way around the tree trunk. Vascular cambium is making secondary xylem toward the inside, and it's making, see, where is it? Secondary xylem, wood. Vascular cambium is making secondary phloem toward the outside, see? And then the cork cambium, that lateral meristem cork cambium, is making phelloderm toward the middle, toward the inside, and cork toward the outside. Hopefully that somehow helped you get oriented. If not, these are some very rudimentary ones I tried to make myself to make it a little clearer for you using the colors I've been using. So lateral meristems, cork cambium, vascular cambium. The lateral meristem cork cambium makes cork or phellum toward the outside, phelloderm toward the inside, both of which are colored in blue here. But as I mentioned, you can say all of these things together are the paraderm, which is a term you might see in lab if you happen to be in lab. Vascular cambium, again, said this a bunch of times, but makes secondary phloem toward the outside, and then it makes secondary xylem toward the inside. And when you are looking at a mature tree trunk, the majority of what you're looking at is secondary xylem. You may have heard of somebody say before, you can tell how old a plant is by how many rings they're inside. So the secondary xylem grows in because the vascular cambium is a ring, it's going to grow each year like this. Like this is how the vascular cambium is growing, wider and wider ring. And when that's happening, it's making the secondary xylem toward the inside. So as it makes secondary xylem toward the inside, it's getting wider, and it's going to be making secondary phloem toward the outside. So each year, the vascular cambium is going to make a ring of secondary xylem. So like, okay, it's spring, now the tree is going to grow. And as the vascular cambium grows, it's going to make secondary xylem. So, whoop, okay, I made it. So that's what this is trying to show you here. So let's see if I color coded it. Yeah, so where is that? So look here. This is better to show you on this one. I'm going to use green because it's just, actually, no, I'll use the same. Look, secondary xylem, sorry, vascular cambium, right in a ring. Each year it's growing the secondary xylem toward the inside, right? Toward the inside. And so look, this year it's still actively growing, but last year it made this. Last year it made this one ring. As it keeps growing, you'll make another ring, etc. And you can tell how old a plant is, give or take, that how old the trunk is. There's a question, what does it mean when there's a gap between the rings? There won't be a gap between the rings. There won't be a gap between the rings of the secondary xylem. I'll show you though why it might look like that, which is on this next slide, which is showing you each year, each picture, okay, in the fall, you know, the trees kind of go to sleep a little bit in the fall, right? The leaves fall off. They don't really grow or anything during the winter time, most of them. They kind of just like hang out. But then spring comes. And with spring, what? April showers brings May flowers, right? So like when spring comes, a ton of rain will fall. And that rain will allow a lot of growth to occur in a very short period of time. And so cells that are made during the cells of the secondary xylem that are made during the spring tend to be much bigger. Then summer comes, there's a lot less rain, for example. And so the cells of the secondary xylem in the summer tend to be smaller. So I'm mentioning that because that's what's saying here. So it produces annual rings, right? So this is secondary xylem tissue. Here is one annual ring, but that ring shows both the early and late wood, or spring wood and summer wood. So what color would be good to use? I wish I had more colors. I'll use pink, right? So see how the cells here in this tissue are wider, right? Like one circle is wider than the cells located in here. Like do you see how the ones, you see how the cells here are wider than the ones here? That's because the cells here were made in spring, so they grew faster with all that rain. And the cells here grew in summer with less rain, so they're smaller. So one annual ring will consist of both that spring wood and summer wood. So that could, depending on the cut of what you're looking at, it could look like gaps. Here's a real example, like a super real example. So in this example, it's easy to see the spring and summer wood because the spring wood is lighter in color and the summer wood is darker brown. So look here. So see this? All right, what's the best place to start? I'm gonna kind of work in this area here because it's the easiest to kind of annotate. So see this darker area? that would be summer wood. And see this lighter area? That would be spring wood. And so if you wanted to count how old this tree was, in this example, I would just count all the spring wood because it makes it easier and they're more visible. So like each of these lines, 1, 2, 3, each of these is an annual ring of, well, hold on. Oops, right. Summer wood, spring wood. So each year, I should do it the other way. Each year, spring wood, summer wood. Next year, spring wood, summer wood. Next year, spring wood, etc. And so you can count that, right? 1, 2, 3, 4, 5. Each of those is a year of growth and tell how old. It's amazing how you identify spring and summer wood. Good. Okay. All right. Next here, I think we're getting close to the end here. Yeah, so this is showing you the same thing before, like you see the spring and summer wood. I'll use a different color. I'll use yellow, right? So this is that spring wood. This is that summer wood for this annual ring, which is shown in blue. You might also notice though, when you're looking at a cut of wood, that you might see these kind of long things kind of radiating from the center of the wood out to the outside of the wood. And those are things that are called wood rays, and they help move things across from the inside to the outside of the stem. So when I say move things, I mean water and minerals. Let's look at it on a real piece of wood, right? On this real piece of wood, I'll use pink for the annual rings, right? So it's a little harder to see the annual rings on this wood, but like here's one annual ring, there's another one there. There's many, right? Like here's one ring, right? There's many annual rings. But in addition to those annual rings, you see these almost looks like bicycle spokes, right? Spokes of a bicycle wheel going from the inside of the stem to the outside of the stem. And those are those wood rays that help move water and nutrients from the inside to the outside of the stem. Going back to the last slide, does the dot in the middle count as spring wood? So do you mean probably this one? Yep, yeah. So that dot in the middle is that first year. That's a good catch, Crystal. Like that's really, so that little dot in the middle that Crystal saw, see that? That dot in the middle would have been the first year that stem was able to get woody. So that would have been the very first year that it was able to do secondary growth. And it would have started it in the spring. So that lighter colored dot in the middle is that very first year of spring wood for that tree. Good catch. Good. No, that's good. And in terms of the other things we talked about today, I gotta use a different color, but like out here, this is where you would have that cork cambium. I guess I will try to use the same color because it's hard to see, but like the cork cambium would be out here. And the cork, remember, so like this, see all this stuff that you call bark, right? That's the cork. And then the phylloderm would be made toward the inside of the stem out there. Okay, got another question. Oops. Can you tell the age of a tree by the growth? Yes, yes, you can. You can tell the age of the tree by, you can estimate the age of a tree by counting the rings. Yep, you could also estimate the age of a twig by counting things too. But I don't know if we have time to talk about that today, or maybe I do in a subsequent slide. All right. Here, this is another microscope slide just showing you basically, if we were to take this, we would take this and look at it under the microscope slide. All you would see would be secondary xylem, AKA wood. But even there, you can see the rings, right? Here, this is good because you can see the spring and summer wood. So look, let's take this one. So look, here's a ring. And then It's a little hard to see how it's cut, but like here's another ring. And look. I'll use green. Notice how big and wide the cells are there, because that's the spring wood. Notice how small and puny, right? Like right there. The summer wood xylem cells are, right? Look there. Look how small and compact that is compared to that, right? Much bigger. And look, Crystal, there you go. There's your first year of spring wood right in the middle, right? Look how big, look how fast it was growing. All right. I don't care about resin ducts. Oh, yeah, no annual rings, right? See the top, no annual rings. So once again, here's an annual ring. Here's an annual ring, right? Here's an annual ring. And it's a little harder to see because this is a young tree, but this is all the spring wood, and then that summer wood is just that little area there. Did most of its growing in the spring. Yep, good. All right, let's see where we're at here. Oops, get out of here. I think all we have left is the, oh boy. Okay, let's see here. 7:22. I think we can finish this. We can finish this. All right. All the things I just talked about on the previous slides, right? So like, for example, when you're looking at a tree like this is one orientation that you can look at a tree, right? If you cut the tree down, this is what you'll see. But again, if you've ever bought a 2x4, you can cut wood in lots of different ways, right? Like to make a 2x4, you're not cutting the tree like this, you're cutting it down like this first, and then you're kind of shading the inside of the trunk to be in the shape of a 2x4. So depending on how you actually cut the tree trunk up, you'll be able to see different views of the different cells that I've already talked about today. So for example, look, this is a wood block, right? Like a block of wood, right? And depending on what side of the block of wood you look at, you can see different structures, right? So for the way that we had been looking at it, for example, this way, right? You would see the structures would look like this. Would look like this view, right? So for example, an annual ring, and then like the cells of the xylem and xylem cells kind of looking like a straw. Look, here's a wood ray radiating out from the center of the stem. But if you cut the wood in different ways, you're going to see it you're going to see these structures in different views. And for any of you who are into carpentry or doing things like that, where you care about things like wood grain, these are things that you would consider, because you might want certain things to be visible or not, depending on what you're trying to achieve. I don't want to spend a ton of time on this. I'll just go forward, right? This is the same thing, but in a different way of drawing. So this is again, oh, so this says transverse. Don't worry too much about that. I'm just trying to show you that depending on how you cut the wood, you see things in it differently, right? Differently. So this is, I don't really care too much about this. This is kind of a little bit better, right? Like this is what I really want you to kind of get from this. Look, so when we cut the tree down, right? Cross section or transverse section, it's all the things I already talked about. Annual rings of spring and summer wood, right? Wood rays, right? But if you cut the wood like shown over here or shown over here, you'll see different structures, different views of it. And why that's interesting is because, look, there's a lot of different types of wood. This is just an example of some types. And depending on the species of tree, there's a lot of variation in color and the how close together the annual rings are, how far far apart the annual rings are. What I like about this slide too is it kind of gives you an idea of like what we use these things to do. So for example, this hard maple we use to make bowling pins. Bald cypress we use to make railroad tracks. Red spruce we use to make violins. Oak we use to make barrels, maybe for any of you knowing about like alcoholic beverages, you sometimes use oak barrels, right? From those of you who've been to Cooperstown, right? Ash trees to make bats. So, but it's kind of neat to look at the different variations, right? Like look how pretty hickory is, like kind of chocolate vibe, whereas like the bald cypress is more red. On the white oak, you could really see those wood rays, whereas like you don't see any wood rays really here or here. So just kind of interesting to look at the different types of wood. Okay, heartwood and sapwood. So it's not complicated. Basically, like once you get to be a tree that's like this big, that's me in a park in Queen with a tree. This was an ash tree that had been cut down because of the emerald ash borer. But like, and I counted out this tree. I think this tree I aged to be about 150 years old, give or take. But once you are a tree this big, right? So like, look at all this trunk, and that's all secondary xylem. You don't need all of it to be functioning. You don't need all of those cells to be moving water. It's too many. And if anything, what you really need is structural support because you're giant, right? You're giant. And so that's what heartwood and sapwood refers to. Heartwood, and this is shown in kind of a darker color, is secondary xylem, heartwood, secondary xylem that no longer functions in moving water. Whereas sapwood, which is shown in kind of lighter color here, is secondary xylem that still functions in moving water. This is a different version of that from the side. So this heartwood in the middle here, use different color. This heartwood in the middle here is now really serving almost like a scaffolding or like pylon, right? Like it's literally just a structural support. Whereas the sapwood is still making water go from the roots to the top of the tree. Why I like to include this is because some people like to carve into trees because of different reasons. And I'm just letting you know that for a really big tree, the majority of the inside of the tree is heartwood, which means not functioning anymore. It's only serving for structural support. Whereas the living part of the tree, sometimes for a tree that's like 6 feet wide, the living part of that trunk is only a few inches under the surface. So by carving into trees or nailing things into trees, you can actually cause pretty significant damage, even if you think you're only brushing the surface, because you're actually not. Yep, yeah, okay. Yeah, so don't carve into trees. Don't carve into trees. Okay, look at this. Wow, so look, sometimes trees can get so much secondary growth that you can actually like, so by the way, this tree is dead. By the way, this tree is not alive. So that's why they're able to do something like this. If you did this to a living tree, it would die, which is, I'm not sure if it died before or after, but yeah. But anyways, look at, so that's what I'm saying. It's the structural support. That's a lot of, is there a way to find the age of a tree by not cutting it open? So you can, there's a whole field, I'll write it in the dendrology. Dendrology is actually a study of aging trees. Believe it or not, that's a thing. And dendrologists, and they're actually called dendrochronologists, are the people who study dendrology and the people who age trees. And they actually have a very, very thin borer, super thin, that they can go into the tree and pull out a section that's like the size of a straw and use that to age the tree, and it won't cause enough damage to hurt the tree. You do have to, at the end though, seal it up so that insects can't enter or fungus can't enter. Okay. Stems is so 75. Okay, we are so close. Oh gosh, we're so close. We're so close. We're almost there. Hang in, hang in. I thought about taking a break, but literally there's 10 slides left. So let's just power through. We got this. We got this. Maybe just a moment of me not talking for a second. Oh. This is harder than it looks, all right? I'm telling you. All right. So we talked about growth, primary growth, secondary growth. We talked about mature stems. Now let's talk about stems of dicots and monocots. So first I have to tell you what dicots and monocots even are. So in the flowering plants, which we haven't covered yet, but we will, there are flowering plants that are called dicots, and there are flowering plants that are called monocots. We'll learn more about them in a later lecture, but their stems are different. So we have to talk about them today because we're talking about stems. So the dicots have about 200,000 species, give or take. It's a very diverse group, and some of the ones you may be familiar with are cacti, anemone, poppies. There's a lot of different types of dicots, though. They're the largest of the group. Like in the flowering plants, most flowering plants are dicots. Monocots are less common. There's only about 90,000 species of monocots, but some of them are very important to us. For example, grasses, and you might remember last time that grasses make up a lot of our food, right? Wheat, rice, corn, those are all grasses. So in addition to that, palms and banana trees, rice should just be in grass, sorry. Rice is a grass. But yeah, orchids, cattails, etc. are examples of monocots. There's other reasons why we have these groups, monocots and dicots, which we will talk about during the angiosperm lecture. But in the meantime, since we're talking about stems, it's just important for you to know that dicots and monocots have different arrangements inside of their stems. Yes, rice is a grass. We covered that last time, Haley. Rice is a grass, wheat is a grass, corn is a grass. Yes. Okay, so vascular bundles, which are areas of those vascular tissue, xylem and phloem, are arranged differently in dicots and monocots. So if you were to do a cross section, and the way I like to just summarize it so it's easy. So if you're looking at a dicot stem, the vascular bundles are arranged in a ring. When I say vascular bundle, I'm saying the areas where primary xylem and primary phloem are located. That's what I mean by that. This is only about primary stuff. Whereas in monocots, the vascular bundles are scattered around. So let's show you what I mean by that. So let's use pink for dicots. So here's dicots. Vascular bundles arranged in a ring. Here's a vascular bundle. Here's a vascular bundle. Each of those has primary xylem, primary phloem inside. So here's another vascular bundle with primary xylem, primary phloem inside. Look, here's another vascular bundle, primary xylem, primary phloem inside. Here's another one. Here's another one. I don't need to draw them all, but you should be able to see that the vascular bundles here are arranged in a ring. So that's what they look like in a dicot stem. Monocot stem, vascular bundles are scattered. So each of these little dots on this monocot stem is a vascular bundle. And notice, they're not arranged in a ring. They are scattered. So this is A monocot stem, right? Notice scattered. Each vascular bundle is scattered around, right? Each of these, not arranged in a ring, all scattered. One cool thing though, I always think that this kind of looks like a little face, but if you were to look close up at one of the vascular bundles, this is what I was trying to tell you, is like you have your primary phloem and you have your primary xylem there. It's okay if you don't really see what I'm talking about. Just know that this region here is your primary phloem and this region here is primary xylem. Yeah. Notice though, don't worry about noticing. That's okay. You don't see. And then here, dicot stem. So vascular bundles in a monocot stem, scattered. Vascular bundles in a dicot stem, arranged in a ring. If you were to look at them up close, right? Here's your ring of them. Look up close. You have your xylem tissue toward the inside, phloem tissue toward the outside. I am specifically talking about primary. You don't get confused here because you do already see the vascular cambium. This stem is obviously on its way from transitioning to from primary growth to secondary growth. And so we already see a vascular cambium here, but notice we still see an epidermis. We still see a cortex. We still see a pith. But I just don't want you to be confused about why you see vascular cambium there. It's because it's on its way to becoming secondary growth already. Right. All right. Another thing, so monocots and dicots have different arrangement of vascular bundles. And in terms of secondary growth, dicots can make the species. Plants that are dicots can actually do secondary growth, which is again why you're starting to see the secondary structures here with that vascular cambium. But monocots rarely do secondary growth. So for example, back here, right? Monocots, you have lilies, grasses, etc. Those are not things that make a lot of secondary growth. Here's bamboo, for example, which is also a type of, which is also a monocot. And bamboo is a grass too, just so you know. Bamboo is also a grass. But yeah, so look here on the left, you have dicots. Dicots are going to be your things that make what, wood, real wood, via vascular cambium. Monocots might get a little more thick, but they're not going to make that large amount of secondary xylem like a woody dicot look. All right, this is the last part and it's super fast. Okay, some plants have special stems that let the plant do something a little different. So we say that that plant has adapted to live in a certain environment with these by having these modified stems. So for example, on the left here are some examples of modified stems. You have these two on the top. This is zero, use different color. You have a rhizome, which basically is a thick stem that grows underground and lets the plant spread to new areas via that stem. You have a tuber, which allows for food storage. If you've ever seen a grapevine grow, you may have seen something like this that's called a stem tendril, which allows the grapevine to climb. I should keep that there. You also have a stem that's called a runner. A runner is similar to a rhizome in that basically it allows the plant to spread to new areas using its stem. Then you have something like a bulb, for example, an onion. So an onion, believe it or not, the base of the onion, that thing you cut off that you don't want to eat, is the stem of the onion. And the other part of the onion that you like to eat is actually leaves, believe it or not. But this stem here helps to regulate food storage. Yeah, you don't need to know more about that. Corm is another one that both corm and bulb, I don't know if you have a garden or if you've ever planted a bulb, like a daffodil or a dahlia, the advantage of a corm or a bulb is that during the winter time, everything above the ground can die. And then that little bulb or that little corm will stay under the ground and be frozen. And that's a stem. And then when spring comes, it's able to like emerge again. So it lets plants survive the winter that normally wouldn't survive the winter. Last example of an adaptation are thorns, right? So some stems have thorns. This here is a stem, believe it or not. That's a stem, and this stem has thorns on it. Okay. We did it. Oh, we did it. A little bit longer than I was expecting, but we did get through it. All right, guys. Well, I hope you're awake. Thank you for being with me today during this live lecture. Quiz number 2 is available on, it's probably going to be available in 20 minutes or so on Canvas, but I recommend, this is a lot of new information today, right? So don't rush. You have until Wednesday by 6 p.m. to submit the quiz. You just did a quiz for this first lecture, so you get a sense now of the type of questions that I ask and the level of difficulty, right? So use that knowledge to prepare for the second quiz. Don't rush it. Take your time. Remember, once you have only one attempt at the quiz, but the quizzes are not timed. So you can take as much time as you want to do that. Thank you, everyone. And then, yeah, next time we will talk about the leaf and the root. If anyone has any information or questions right now, I'm happy to answer them. If not, send me an email and I will follow up with you there. Also, don't forget about those discussion boards. Yeah, thanks for hanging in there. I know this one was a lot of information. All right, guys. Have a good one.